Good afternoon, everyone, and thanks for joining us again for our Occ-COVID Science Solutions and Success Stories. I'm pinch hitting Kimberly O'Connell here for one of my colleagues today who put this session together, and Val and I are co-hosting you today to introduce our first speaker. Val will then talk a little bit about the Global Summit and pandemics when Raymond has finished his chat. I did also want to mention that please do revisit the webinar series, and you can, all of our archived videos since last July around COVID are there for you to share with your joint health and safety committees, with your health and safety meetings, and it's really great content. And Raymond was a very significant part of that earlier in January with this session on erring on the side of caution. And we are planning another for August 20th, which will be recorded as this one is, that I wanted to let you know about. So without further ado, I'd like to introduce you to Dr. Raymond Tellier, who again has been with us in this series before. So Dr. Tellier obtained a BSc in physics from the Université de Montréal and an MSc in mathematics from McGill University. He then obtained a medical degree and an MSc in microbiology from the Université de Sherbrooke. He did his internship at McGill University and his residency in medical microbiology at the University of Toronto. Then did postdoctoral studies at the National Institute of Health in the hepatitis viruses section. Dr. Tellier worked as a medical microbiologist for 12 years at the Hospital for Sick Children and the University of Toronto. He was a medical microbiologist for 10 years at the Laboratory for Public Health of Alberta in Calgary, and a member of the faculty at the Cummings School of Medicine in the University of Calgary. He's currently medical microbiologist at McGill University Health Center and associate professor at the Division of Infectious Diseases, Department of Medicine of McGill University. Dr. Tellier has published 112 peer-reviewed scientific articles. His research interests include molecular diagnostic methods in virology, hepatitis viruses, respiratory viruses, opportunistic viral infections of the central nervous system, and emerging viruses. And he's here to speak to us about vaccines and variants today. So welcome, Dr. Tellier. Thank you.
Thank you very much for this kind introduction. And there we are. All right. So as we know, what we're dealing with for the past year and a half is a coronavirus. Coronavirus is a very large family of viruses. There are many coronavirus infecting several different animal species and also humans. And you can see here within the subfamily Coronavirinae, they are grouped into 4 genera, which are the alpha, beta, gamma, and delta coronaviruses. And human coronaviruses, we know that there are at least 4 of them that have been with us for a very, very long time that are causing a very frequent infection and which manifest as common colds. There were very poetic names such as OC43, HKU1, etc. We have been dealing also with emerging coronaviruses, and this came as a big shock in 2003 with the example of SARS, which afflicted Canada was one of the afflicted countries, and this was a very dangerous virus, which fortunately was contained and didn't give rise to a pandemic. And then later on, we had the emergence in the Middle East of another coronavirus, which is called the Middle Eastern Respiratory Syndrome coronavirus, which is, thank God, poorly communicative, but does not transmit well between humans, but is associated with a very, very high mortality of about 39%. So a very worrisome virus. And of course, for the last year and a half or so, we have been dealing with a worldwide pandemic, which is still ongoing, caused by a virus called SARS coronavirus 2, which is indeed a close cousin of the SARS coronavirus 1 that was the causal agent of SARS in 2003. So this is just a schematic which show you the different grouping of the coronaviruses. This is the beta coronavirus here, which is further divided in subgroup. So in the subgroup 2A, this is where you have the OC43, which is one of the coronavirus causing a common cold. And in the group 2C over here, this is where MERS here. In the subgroup 2B is where we find the agent of COVID-19 and the agent of SARS, as well as a very large collection of closely related bat coronaviruses. There's plenty more where that come from. And of course, the concern is that these viruses, which would include the ancestor of the COVID virus, there are many other viruses that have a potential, we think, to emerge. This is a schematic of a coronavirus. So it's an envelope virus. It acquired a membrane from a cellular membrane from the cell from which it will bud. The RNA virus is coiled with the nucleoprotein inside, and you can see embedded in the membrane a surface glycoprotein that is known as the spike, which has a prominent head here. And this gives the appearance to these viruses of a crown with the string of pearls around the crown, hence the name coronavirus, because corona in Latin means crown. And the spike glycoprotein is going to occupy a very, very central place in our discussion today. These are electron microscopy picture of different coronaviruses. So this is a picture that was taken at the British Columbia Center for Disease Control during the SARS outbreak. So this is a SARS coronavirus, the agent of SARS. This is the agent of the MERS coronavirus. So you can see that these viruses are circular with a kind of string of pearls around them, which give the appearance of the virus of that of a crown. This is an electron microscope picture of the SARS coronavirus 2, the agent of COVID-19. I don't know why, but it is now the fashion to colorize these pictures, which to me is a little bit like colorizing the movies in black and white, like Casablanca. I am not sure it's a good idea, but this is how it's done nowadays. Electron microscope is inherently giving picture in black and white. We now understand very much better than in 2003 during SARS the natural history of these viruses. We know that all the coronaviruses that are infecting humans came to us from an animal reservoir, usually by transiting through an intermediate species before coming to humans. So for example, for the OC43, the original host was a rat, and it came to us through cattle, through a virus that would have been very similar to the bovine coronavirus. Most of the nature's reservoir of coronavirus are among bat species, as you can see. There's also no reason to believe that an intermediate species is absolutely essential. It could be, in theory, quite possible to have a virus jumping directly from bats to human, and it is perhaps what has happened with the COVID coronavirus. These things have been going on for a long time. This comes from a paper that was looking at the genetic sequence of the OC43 coronaviruses, and by using genetic analysis and molecular clock, they timed the introduction of this virus into the human species sometime at the end of the 19th century. And that's very interesting because in 1890, there was indeed a worldwide pandemic, which was attributed to influenza, but there's now a rethinking that perhaps it was not influenza, but it would have been the introduction into human societies, into human species of these coronaviruses that eventually quieted down a bit to become the OC43. So to borrow a line from one of my favorite science fiction TV show, Battlestar Galactica, these things have happened before and will happen again. And so what we're seeing with the COVID pandemic is a very important example of this phenomenon of a virus emerging in the human population, of a coronavirus emerging, and we know that they like to do that. And even though right now in Canada And in Quebec, where I'm speaking from today, we are in a rather good position because the number of cases have greatly diminished and the vaccination has been increasing. But from a global perspective, in most places in the world, it's never been that bad. So this pandemic is far, far from being over as we speak. So this is again a schematic representation of a coronavirus, of the SARS coronavirus 2, as it were. And the spike glycoprotein that you see here, one of its main function is to attach to a cellular receptor on the human cells, which is the ACE2 receptor, so the virus will bind, and this will initiate the process of the entry of the viral genome into the cell, followed by the synthesis of the viral protein, the replication of the genome, the assembly of new viruses, and the infection will then continue that route. And as it turns out, and you can see here another representation of the spike glycoprotein with a sub-region within the glycoprotein that's known as the RBD, the receptor binding domain, which is the part of the protein that will bind to the ACE2 receptor. And this protein has a very important function, as I've just explained. It is also the target of a most neutralizing antibody. And all the vaccines currently licensed or being developed include at least the spike glycoprotein as an antigen. And in fact, most viruses contain only the spike glycoprotein or a subdomain of the spike glycoprotein as the viral antigen to elicit an immune reaction that would be protective. So this is a very, very important protein in the story that is unfolding. The massive nature of this pandemic has triggered an unprecedented effort at vaccination, massive vaccine development. And as of January 2021, there were more than 170 vaccines that were at various stages of development, 63 of which were in human clinical trials. And basically, every techniques essentially that have been thought of to develop vaccines are being tried. We're throwing everything in the kitchen sink to make vaccine about this, including mRNA vaccine, vector-based vaccine, DNA vaccine, inactivate killed virus vaccine, live attenuated vaccine, cloned protein or virus-like particles, etc., etc., etc. So obviously, I'm not going to talk today about all of these. We don't have the time to do that, and most of them are very early in the development. We will concentrate on the 4 vaccines that have been authorized in Canada. 2 of them use the mRNA vaccine strategy, which has the Pfizer-BioNTech vaccine and the Moderna vaccine. And the 2 others, the AstraZeneca and the Johnson & Johnson, are a vaccine based on using a crippled adenovirus vector. I'm not going to talk a lot of the Johnson & Johnson because although it's licensed in Canada for a variety of reasons, it has not been used much so far. So I'll really concentrate on the first 3 vaccines. So this is a schematic for to illustrate how an mRNA vaccine works. So what you do is that you synthesize a RNA, a messenger RNA that codes for the antigen of interest. In this case, it codes for the spike glycoprotein or for a subdomain of the spike glycoprotein, depending on the vaccine. And it is embedded in lipid nanoparticles that protect it against the environment and will also allow the mRNA to enter the cell through fusion of the lipid with the envelope. And then the messenger RNA is going to be translated by the cell into the viral protein, into the antigen. That spike glycoprotein will emerge at the surface of the cell, where it will be spotted by cells on the immune system, by B lymphocytes, which will trigger the formation of antibody against the spike. This is what you want to do. Also, inside the cells, some of these glycoprotein are going to be cleaved into small peptide that are then presented outside the cells by some specialized protein at the surface of the cell to other member of the immune system, so to T lymphocyte, which will reinforce the synthesis of antibody and also trigger other kind of immune reactions, cellular mediate immunity. So this is a rather complex immune reaction, a very complete immune reaction that is triggered by this way of doing vaccination. It's the first time that we are having commercial vaccine with messenger RNA technology. However, it's not as new as you think because these things have been in the works for many, many years, more than a decade, in fact. And they were, so all the details about the technology of the lipid to preserve the mRNA to enter in the cells and their safety in terms of toxicology and etc. All these things have been worked out for years already. And they were being studied to design some vaccine against other viruses. For example, some of the scientists that are part of Moderna, they were working before the pandemic on developing with that technology a vaccine against the respiratory syncytial virus. And when the pandemic came out, they basically redirected the technology and all the basic knowledge that we had to do that. And in a way, I'd like to argue that in its principle, it's also not as novel as you would think, because if you think about one of the oldest methods of making a vaccine, live attenuated vaccine, like the measles vaccine or the Sabin vaccine against polio, what you would have is a virus that's attenuated, so not very pathogenic, infecting the cells. The messenger, it would then synthesize its messenger RNA, and the messenger RNA would synthesize a viral protein, which would then be presented. So this way of doing a messenger RNA vaccine instead of a live attenuated vaccine, you cut the middleman from the process, or rather the middle virus. You zoom in right away to the mRNA and synthesis of the protein. So in clinical trials, the two mRNA vaccines against COVID have shown extremely high efficacy. So 95% efficacy in preventing illness in the stage 3 clinical trial for Pfizer, and very similar numbers for Moderna. Okay, so this was in preventing illness. And studies in the field have essentially confirmed these remarkably good numbers. And so in the studies that were done when the vaccine was introduced shows that for the Pfizer, you had an efficacy of 91% against infection. So that's not just disease, it's against infection as verified by PCR. So these people do not become infected at a 91% efficacy level, meaning that they're not only protected against disease, most of them are in fact never become contagious. Okay. And there is also, it is however not 100%. So you will have a small number of vaccinee that would have breakthrough infection. But the studies that have been done looking at the breakthrough infection, they found that the illness was much milder, and that the level of virus that were found in these people, the viral load was considerably reduced, so that in breakthrough infection in vaccinated people, they were much less contagious. This is a slide that's another very extensive study that was not done in Israel. So Israel is really the country that has achieved the highest level of vaccination in its population the fastest, and they did that using only the Pfizer vaccine. So this has enabled to do a very large-scale study of this vaccine. And what you show here is two curves of various markers in two populations, the vaccinated population in blue and the unvaccinated control population in orange. And this is days after the first dose. So you can see that both groups become infected at about the same rate for a few days following vaccination. And then at some point, around 14 days or so, the immune reaction triggered by the vaccine starts to kick in action. And then you see that the people that have been vaccinated, their rate of infection is essentially drop off and they stop being infected, whereas the control group continue to be infected. If you look at infection with symptoms, it's the same story. You have an even greater difference if you look at symptoms severe enough to warrant hospitalization or transfer in intensive care unit or death. So this is a remarkably convincing illustration of the efficacy of this mRNA vaccine in real life to prevent infection, to prevent severe disease, and to prevent death. And again, what they could verify is that even though the efficacy of this virus is not 100%, even for those that do have a breakthrough infection, the viral load that they have is much lower than in the unvaccinated people. So they would be the small proportion of breakthrough infection, they will not be as contagious as people that are infected without the benefit of having received the vaccine. The side effects, so this is a slide that summarizes side effects for several vaccines, but if you look at the number 1 and the number 2 that are illustrated here, which are for the Pfizer and the Moderna vaccine, you can see that they have roughly the same incidence of side effects that are mild to moderate, which are pain at the injection site, redness, swollen lymph node, fever, fatigue, etc. So these are all symptoms that were mild to moderate that are quite common among people receiving the vaccine, as many of you who have received these vaccines can attest. These are of short duration, a few days, and they are self-resolving. And this is not at all unexpected for vaccine. There are many other vaccines that give similar side effects. There has been a more serious side effect with vaccines. So we have seen initially with the Pfizer, but also with the Moderna, there has been a very rare incidence of anaphylactic reaction, so severe allergic reaction that warrant immediate treatment. It has remained quite rare. So the estimate for the Pfizer are around 11 cases per million dose and 2.5 cases per million dose for the Moderna vaccine. It's a little bit higher than what you see for other vaccines. For example, for influenza, you have 1.35 anaphylaxis per million dose, but it remains a very rare complication that has remained eminently manageable. All these cases of anaphylaxis were treated successfully and resolved without death and without sequelae. So no one to date has died of anaphylaxis in response to the vaccine. It is, however, the reason why when you receive the vaccine, you are asked to stay on the premises for at least 15 minutes just to make sure that people don't develop an anaphylaxis reaction. And it also means that in vaccination centers, you must have the medication and the setup to treat an anaphylactic reaction if it were to occur. Very recently, there has been a report of another very rare reaction, which so far has been noted only with the Pfizer vaccine, but probably we'll see it with the Moderna, when the number of vaccination with Moderna catch up, which is instances of myocarditis, which is an inflammation of the cardiac muscle. So this was not seen for many, many months into the vaccination, and it's when they moved the vaccination to a lower age group that it was noticed, first in Israel, but it has also been reported in the United States. The link with the vaccine is not completely clear because we're talking about a very, very low percentage, about 10 per million or a little less than that. And myocarditis occur at a very low frequency in the population in general for other reasons. So it's not completely clear that it is linked with the vaccine, and it's currently being monitored. All the cases that have been reported shortly after vaccination, they've all recovered with treatment, which is for mild myocarditis, which all these cases were. The treatment is just non-steroidal anti-inflammatory drug, and they all have recovered without sequelae. So that does not constitute an obstacle to vaccination, especially if you remember that myocarditis is one of the complications of COVID itself. And COVID is associated right now with a 2% mortality and with a great many sequelae. So if you look at all of this, the benefit and risk equation remain overwhelmingly in favor of the vaccination. To move on, I'd like to talk to you about the AstraZeneca and Johnson & Johnson virus. So the strategy of vaccination is to use a crippled adenovirus vector. So this time what you use is the clone gene for the spike glycoprotein inside an adenovirus vector. The adenovirus vector will infect the cell, inject the DNA in the nucleus where it's transcribed into the messenger RNA. The messenger RNA goes in the cell, translate the spike glycoprotein, and you have the same presentation of the antigen to the immune system as we saw before. The adenovirus vector has been engineered in a way such that it is crippled. It doesn't have all the gene that the adenovirus needs to replicate itself. So it's an abortive infection. The viruses that you give in the vaccine enter the cell, deliver their message, and then cannot go and manufacture more virus. So that remains, that does not proceed any further. A theoretical advantage of an adenovirus vector vaccine is that because the adenovirus structure would trigger innate immunity, which is synergetic with the adaptive immunity, theoretically, it should produce a better immune reaction. That was the hope anyway. Other advantage for these vaccines is that they're very cheap to produce and they could be produced with a more standard production plant, whereas an messenger RNA, which are a new technology, requires dedicated manufacturing capacity, which early on in the pandemic was a bit of a bottleneck. These viruses also are very stable. These vaccines are stable if they're kept in an ordinary refrigerator, whereas the messenger RNA required freezing at -20. And early on, it was thought that the Pfizer required, in fact, deep freezing at -80. So that was making the logistic of distribution more difficult. A theoretical objection to the adenovirus vector is that adenovirus infection are not rare in the human population, and so pre-existing antibody against the vector might dampen the effect of the vaccine. And this has been countered in a number of ways. So for the Johnson & Johnson vaccine, they chose a crippled adenovirus 26, which is a relatively rare type of adenovirus, so that most people would not have pre-existing immunity. And the AstraZeneca was engineered around a chimpanzee adenovirus to which none of us have in principle been exposed. But you remain with another objection, which is once you've used a type of vector for a vaccine, you probably cannot use with the same efficiency that same vector for another vaccine against another virus. So that is something that time will tell. So the clinical trial for the AstraZeneca showed that the efficacy to protect against infection as detected by PCR was very good, although not quite as high as the 90% or so seen with the messenger RNA. The protection against severe disease, however, was as good as the messenger RNA. It has also been pointed out by many observers that it's very difficult to compare these clinical trials because the population were not quite the same, and by the time that the AstraZeneca had moved to clinical trial, there were already some variants that were in the mix, which might complicate a little bit the statistics. Be that as it may, it was a virus deemed very efficacious, very efficient in clinical trial. And this has hold on in trial in the field. So in the United Kingdom, where the vaccine has been used at a very, very high level, studies have shown that after one dose of either the Pfizer or the AstraZeneca, you had an 80% protection against severe disease, and that with two doses, you have close to 100% protection against severe disease. And the author of that studies concluded that there was essentially similar level of protection at the time using either the Pfizer or the AstraZeneca. The side effect, well, I've shown you that slide, but this time we will concentrate on the yellow color, which is the AstraZeneca. And again, you have the same kind of mild to moderate side effect following immediate vaccination. Pain at the injection site, redness, swelling, swollen lymph node, fever, chill, joint pain, etc. And again, like the Pfizer and the Moderna vaccine, these are short-lived. They disappear after a few days with no sequela. There has been also some severe allergic reaction of anaphylaxis that have been noted with AstraZeneca. The European Medicine Agency noted 41 cases in 5 million, which would be an incidence a little bit higher than what was seen with the Pfizer and the Moderna. But again, all these cases were managed with standard treatment for anaphylaxis, and all of them resolved satisfactorily. So there has been no fatalities with that, and that remains also a very manageable and rare complication of this vaccine. What has been very unfortunate, however, is the discovery, as surveillance of vaccination was ongoing, of unusual blood clots. So this is not the usual blood clots that you would be getting with other medication or with deep vein thrombosis. It was a very unusual type of blood clot of thrombosis with thrombocytopenia, with very low level of platelets that were behaving a lot like what you see with the rare syndrome of heparin-induced thrombocytic thrombosis. And what was also very disconcerting is that these rare cases were, however, quite severe, sometimes manifesting at cerebral venous thrombosis, and there have been fatalities with that complication. So that complication remains very rare, thankfully. The incidence, however, has been rising as you monitor more. Initially, it was said, well, it happens once in a million. In Canada, the incidence has been overall 1 in 250,000 doses. And it's a complication that can be a little bit difficult to diagnose, a little bit difficult to treat, and can be severe. You have to keep things in perspective. I suppose in Quebec, we have had one fatality because of a clot caused by the AstraZeneca vaccine, but we have had more than 11,000 fatalities caused by COVID. It remains a very safe and very efficient vaccine. And if it was the only vaccine available, we would be happy with it. But as you can well imagine, the way that the conversation has been going is that it's not the only vaccine available. There are other vaccines, the mRNA vaccine, that are at least as effective as this one and do not seem to have that complication, which, by the way, we don't understand its mechanism completely, but it seems to be immune mediated. Is that in these rare cases, there seems to be antibodies that are triggered that produce platelet aggregation. And it does not appear plausible that these antibodies are triggered by the viral antigen, the spike glycoprotein, because you don't see that with the Pfizer and the Moderna vaccine. So it would remain somehow a rare reaction, perhaps, to the adenovirus vector. And interestingly enough, the same complication has also been noted with Johnson & Johnson vaccine. They can give this very rarely this kind of blood clot with a low level of platelets, and the Johnson & Johnson vaccine also use an adenovirus vector. So this remains under observation, and people are trying to elucidate the mechanism of this complication. But it is one of the thing that has triggered interest in the so-called heterologous prime and boost. If you've received your first dose with one vaccine, can you receive a boost with another vaccine? And in particular, for people who have received a first dose with AstraZeneca, can they be boosted with a Pfizer vaccine or with a Moderna vaccine? And in principle, it should work because, as I have highlighted all along, all these vaccines are using the spike glycoprotein as the antigen. They use the same antigen, so they should be boosting one another. And there has been so far 2 studies in the United Kingdom and in Spain that shows that it is quite safe to do that, although perhaps with a higher incidence of mild side effect at the time of vaccination. And the immune response appeared to be at least as good. And there are immunologists who argue that from a theoretical point of view, a first dose with an adenovirus vector boost by an mRNA should have an immune response that is better than if you had stick to first and second dose with mRNA or first and second dose with adenovirus vector. So this business of mixing the vaccine seems to work very well. And it is, as we know in Canada, it's very much accepted as a way to get your 2 doses of vaccination, and it is increasingly being accepted by other countries, recognizing it as a 2 doses of vaccination. Now, this was all the numbers that I've shown you so far. This was with the original strain of the COVID virus. What is, of course, complicating the issue is that we're having several variants that are come out that are the so-called variants of concern that are more contagious and or more pathogenic and or showing signs that they're beginning to drift away from the immunogenicity of the original vaccine, which gives concern about the possible emergence of viruses that would be immune escape mutant that would not be covered by the current vaccine. So currently, we have 4 variants of concern. The first one, the alpha virus, which was initially identified in the United Kingdom, the beta variant initially from South Africa, the gamma initially from Brazil, and as we all hear in the news, now the delta initially identified in India. And these are all the result of a very natural phenomenon, is that viruses mutate. Viruses with a smaller genome mutate more than viruses with a large genome, and viruses that have an RNA genome mutate a lot more than viruses that have a DNA genome, and the coronavirus have an RNA genome, so they mutate quite a lot. This is what a virus is supposed to do. Although, interestingly enough, the coronaviruses are not the most mutagenic of RNA viruses. They're only mutating as fast as the influenza virus. So influenza virus is mutating a lot faster than coronavirus, and HIV is also mutating a lot faster than influenza or coronaviruses. So, things could be worse, but it is no surprise to see that you're going to have variants that are emerging whose property, whose phenotype is a bit different from the wild type. And the first example of that was the alpha variant, which was significantly drifted away from the prototype virus, and it started to spread very rapidly. So you can see here a representation of the spike glycoprotein, and this is the ACE2 receptor. And there is a mutation here, the N501Y, which is one of the signature mutation of the alpha variant, which increased the affinity between the spike and the ACE2. And that is thought to explain why this virus is more contagious, is that it binds the receptor more efficiently, so it takes less virus to initiate an infection. So the same amount of virus can trigger an infection at a much higher rate because the infectious dose is lower. And what you saw in England is that when it emerged, it not only started outbreaks in the place where it originated, but it spread throughout England in the midst of a very active infection rate by the original virus, and the original virus got displaced. This alpha variant outcompeted the previous virus, which is a sure indication that the new variant has a competitive advantage. And it escaped England and it went all over the world. And certainly for the past few months in Montreal and in Quebec, it has been the dominant variant. And it has been the case also in many other places in the world. These are already old slides. In February, it was predicted that it would become the dominant strain in many states in the United States, which is indeed what has happened. It's no longer true because of the Delta variant, to which we're going to come in a second. It was very interesting to see how the evaluation of this variant evolve in the United Kingdom, because at first they say, well, it's more contagious, but there's no difference in pathogenicity and mortality. And then on further evaluation, okay, so maybe it has increased mortality, but there are limitations in the studies. And then on further evaluation, okay, there's a realistic possibility that it is associated with an increased risk of death. And finally, yeah, it is more pathogenic. It is associated with greater mortality. So you have a variant that is now more contagious and is associated with greater mortality. You have a bit of a similar story with the beta variant, originally in South Africans. So it's a variant that's characterized by a certain number of mutations, including the same N501Y mutation in the spike, and an additional one, which is thought to allow for some slippage from some neutralizing antibody. And you can see that again, it replaced the original variant in South Africa, and it too went to the conquest of the world. It is now found in most countries in the world. And you had a similar story with another variant that emerged initially in Brazil and is now present all over the world, which is the gamma variant. And it has a lot of signature mutation, but it shares two mutations with the South African variant, so the 501 mutation and the 484 mutation. And if you look at laboratory testing of these variants using serum from people that have been vaccinated, you can see that with both AstraZeneca and Pfizer, well, you're making a lot of neutralizing antibody against the wild type. It is not quite as good among variants. So the titer of neutralizing antibody is noticeably smaller. And this is of concern. It's an indication that we're beginning to see a slippage of the virus away from neutralizing antibody provided by the vaccine. However, you still have neutralizing antibody. And furthermore, neutralizing antibody are not the only effective arm of the immune response triggered by the vaccine. So none of that indicates that these would be immune escape mutants. And it is reassuring to see that in the field, the protection conferred by the vaccine holds against these variants. So you can see here, this is a study that was done in Qatar, and you can see that for protection against infection, as determined by PCR, you have the same 90% protection against the alpha variant and against the beta variant. However, the protection's not as good after only one dose. So this is the first time that we're going to encounter a message that I will reiterate many times. You really want to get your two doses. Okay, one dose is not quite enough. And if you look at the protection against severe disease, well, then the number are even better. You achieve essentially complete protection against severe disease for either the alpha variant or the beta variant with the Pfizer vaccine. And there's been similar numbers with the Moderna with the Moderna vaccine, and also against severe illness with the AstraZeneca. The Delta variant is the new kid on the block, and it's a nasty one. It was initially identified in India, but by now it's all over the world. And it has a different set of mutation than the mutation I've told you about, including these two mutations here, which is the mutation, the signature mutation that we're using in the lab to rapidly identify the presence of the Delta variant in sample. And it's even more transmissible than the alpha virus. So now we're getting with a virus that is so contagious that it's going to behave a bit like measles in that when it comes to a place, it's going to quickly find and infect people that are not immune against the virus by either prior illness or vaccination. So, this is changing a lot the equation If we're not vaccinated, it's going to spread. There has been also the same, I'll talk about this a little later, the same business of reduction in the neutralizing titers C in vitro. This is a picture for me. I'll use it in other talks. It's a schematic of the glycoprotein with the mutation that are found in the India variant, but it's just to illustrate uh, where these mutations are. The Delta variant is quickly replacing all the other variant and the wild type. You can see the curve, and it is sparking a fourth wave in many countries, actually. So it is quite remarkable to see the progression of this virus. Anyone who is not vaccinated is going to get it unless they cluster themselves completely. As I was alluded to, you do have a reduction in neutralizing titers. So again, this is with the Pfizer vaccine. So one dose against the wild type, you have neutralizing antibodies for most cases. And at the second dose, it's boosted considerably. You do have a lesser neutralizing titer against different variant, and in yellow here is the Delta variant. But if you boost it, you boost your neutralizing titer. You don't have as much as against the wild type, but you have plenty of neutralizing antibody here. And it's the same thing with the AstraZeneca vaccine, is that against one dose against the variants, not very good, but it boosts quite nicely, and you have plenty of neutralizing antibody. And the good news is that in the field, the protection of these of the vaccine that we have against the Delta variant remains extremely strong. So the efficacy against symptomatic illness with the Pfizer is 88%. It's 67% for AstraZeneca, but if you look at the protection against severe disease requiring hospitalization, you can see that both the Pfizer and the AstraZeneca achieve more than 90% protection against severe disease. But you do need your two doses, however. You really need to have two doses of vaccination. It's even more important with the Delta variant than it was before. And so, in summary, and I'm glad we will have plenty of time for some question and answer here. But this current brief review of the literature, and I have to tell you, I have not reviewed all the papers that are out there. It's not quite my specialty, vaccinology, and I'm beginning to be a bit dizzy when I look at all the papers that are out there. But the selection that I've presented you are the main one, I believe, and the gist of it is that these vaccines that we have licensed in Canada, they are very safe. They reliably trigger an immune response against the virus that provide an excellent protection against illness, and especially against severe illness. It has a very high degree of protection against infection, and even when you have breakthrough infection, the viral load that you have is much lesser. So by vaccinating ourselves, we are protecting ourselves, but we're also protecting the people we come in contact with, family and friends, and for those of us in the healthcare system, our patients. What is good news is that the vaccine that we have are currently excellent against the current variant of concerns that are circulating. Some of the points that remain to be elucidated is how long does the immunity given by these vaccines will last, and will we need at some point a booster? And there are uncertainty about that. I have read one study that says that the immunity will last for years. There's another study done in Israel, I'll bet done in a small number of people, that suggests that we need a booster after 6 months. And oddly enough, Pfizer liked that idea. So we'll see how that evolves. But certainly the vaccine that we have right now, they can be strongly recommended. They are safe and effective. And we should really make an effort to continue our vaccination in Canada. I was distressed to see one modelization by the Public Health Agency of Canada that shows that if we stay at the current level of vaccination, this is what we may see in January, a rate of hospitalization for COVID per 100,000 that would be higher than what we saw last January. So this is again strongly support the proposition that vaccination should be strongly encouraged for personal protection and protection of the population and of the society at large. So this is what I had prepared for you, and I think that we should move on to question period.
Thank you so much, Raymond. What a vast amount of information sharing, which is so appreciated. I know myself, along with others, would be happy to hear that, you know, this science was in the works long before. You know, we sometimes think that this magic happened overnight, and it's so nice to hear that a lot of this was in development for other similar viruses previously in the mRNA vaccines I'm referring to. So please, yes, you will. We will have copies of the presentation along with video later on posted on our website, and you are welcome to raise a hand or unmute if you'd like to speak to the crowd, and also if you would like to write in the chat some of your questions, they are most welcome. Valerie well, you could speak up, Val, but she says, can you explain the concerns and challenges around vaccinating youth and children versus supporting adult distribution around the world?
Yeah,
Oh,
so this is not a big concern, I don't think. What is the case is that currently the vaccine is not licensed for young children because there hasn't been the clinical trials data to show its safety and efficacy. Everybody, these clinical trials are ongoing now. Everybody would be quite shocked that there is a major problems. I can't think of a vaccine that is safe among all the vaccine that we have. I can't think of a vaccine that is safe for adult that is not safe in children. Although they might need a dosage adjustment, and this is one of the main motivation for the clinical trial among the pediatric population. And within that pediatric population, it's subdivided into different groups to see if there's a need for adjustment of the dose. But these clinical trials are ongoing, and although we'll need to see the data to be absolutely sure, but there's a high degree of certainty among the medical community that we will find that these vaccines are safe and effective for the pediatric population also, and this will allow us to protect with vaccination also that part of the population.
thanks, Ray. And I know a lot of the myths to the youth that they're hearing on all this fake news is around fertility. And we've seen as far as I know, I don't know if you've heard, but that's the myth buster that needs to go out. And the messaging that these young kids are getting is that they're having menstrual problems after receiving the vaccine. They're talked about, they're spoken to about fertility issues. So yeah, it would be good to get more science in their hands.
I don't know where that comes from. There is nothing in the vaccine that would interfere with fertility. I know of no vaccine that does that. There's certainly no data to support that contention with the COVID vaccine. So this is just one of these things that started as a rumor, but there is nothing to support that.
Yeah, thank you.
And in fact, one of the motivation also for women of childbearing age should be very strong to get vaccinated, because we do know that pregnancy is a risk factor for severe COVID disease if they become infected during pregnancy. So for young women who are thinking about having children or starting a family, a strong incentive should be to protect themselves before the pregnancy by getting vaccinated.
Great. And we have another question on the lines of our youth around, really, I think people are very much thinking about the return to school. Francois mentions, I might have that wrong, that we keep hearing that children don't have severe outcomes from COVID-19. Would you agree with that conclusion? And then how safe will children be when they go back to school with unvaccinated peers?
Right. So some of that goes beyond my expertise, because these questions become public health. But certainly it is true to say that overall, children have a much milder disease when infected with this virus, which is one of the reasons why people wants to do proper clinical trial on the vaccine before vaccinating the pediatric population, because it is felt that the urgency is not as high as for older cohorts. That being said, we need to understand correctly what is going on here. Uh, Children do not get a severe illness from COVID generally, but it is a quirk of the English language that generally means not particularly. And there are some children that will become very ill with this virus. The majority will not, but if you have millions of children infected, you're going to have a significant numbers that, although a minority, will get severe illness. And this has been seen. Some children that were sick enough to be hospitalized to go to the intensive care unit. And there has been some pediatric death. And also the other thing that we are all very concerned about is that the problem with COVID is not just the mortality or the time that you spend in an ICU, is that up to a third of people that suffer from this infection have long-standing sequelae lasting for months or perhaps for a long period of time. And even though this appears quite rare in children, they have many more years ahead of them, obviously, that people that are older, and so we don't want them to be burdened with sequela for a very long time. So I think we will find that it's well worth protecting children against this infection and to move to vaccination once we're happy with the data for the vaccination trial.
That's great. And to your point, it may feel like a public health issue, but we have a lot of our Ontario workers are educators, and when they're dealing with this population that is potentially unprotected, it's how do we best protect educators as workers in the province? So
Yeah,
to your
as we know, there's been a lot of talk also about doing other things that can give some protection. So, in addition to vaccination, vaccination is quite frankly the most powerful tool that we have. But there are other things that can be done in addition or while awaiting vaccination, which include proper ventilation in the schools. We know now that one of the way by which the virus is propagated is by aerosol, but this can be mitigated by proper ventilation. We know that if everybody wear a surgical mask, it provides protection in two ways. One is that the mask protects the wearer, but perhaps more importantly, it prevents an infected wearer from excreting a large amount of droplets of aerosol. And as we saw in the news, the American Pediatric Association has now recommended that when the children go back to school, they should be wearing masks. And depending on what will happen with the spread in the community, that may be something that we'll have to do in addition to the ventilation while awaiting for the vaccination in the younger group.
Thank you. And we do have a question about the current vaccines, including those that are mixed and matched. So, with adeno and mRNA, or the opposite, show that they're effective against infectivity with the Delta variant. So they're asking about the current
Yeah,
vaccination
so
program.
if you've received two doses, either from a single dose of vaccine or mix and match, does it protect you against becoming non-contagious? The answer is yes to a point. Is that the vaccines are greatly protective against you becoming infected with any amount of the virus. It is not 100%. And so there's a small chance with all the vaccines that you have a breakthrough infection that will be little or not symptomatic, but you will be shedding for a time some virus and you can be contagious. That's a minority of vaccinated patients, but that's not a 0% chance. And with the Delta variant, the margin is not as good as with the wild type. So the majority of people that are vaccinated will not become infected or contagious, even with the Delta. But the majority is not 100%. And so depending on the amount of virus that's circulating in the community, additional precautions may have to be taken, especially if you're interacting with people that are more fragile or cannot be vaccinated. But certainly having a full dose vaccination considerably reduce transmissibility for those that have a breakthrough infection. That remains true even with the Delta, but it is not 100%.
Great. And Val, I'll mute and turn off my camera, and you have a question, you can ask it yourself instead of me reading it if you like.
Right. So, yeah. So the zero COVID elimination strategies. What can I say? In principle, in theory, this is a virus that could be eradicated because it does not cause latent infection and it does not cause chronic infection. You have it and either it kills you or you kill it, and there's no environmental reservoir. And we don't think that you're going to have ongoing reintroduction from an animal reservoir. So in principle, it could be eradicated, but it would take a lot of work. We have effective vaccine, but vaccinating the whole population is a considerable challenge. It has to be done, I would say, because we do not know what else the virus has in its sleeve in terms of obnoxious mutant and variant. To me, it's not a given that there can be a vaccine immune escape virus. For example, the measles virus is also an RNA virus. It keeps mutating. We are now up to 22 genotype or subgenotype, and yet the same vaccine that was designed decades ago is still as good against the newer strain as it was before. However, this is no guarantee either. The COVID virus is a brand new virus. It was unknown to science up to until January 2020 or so. And we do not know its full repertoire of dangerous mutation or perhaps possibility of immune escape mutant. And the longer we let the virus run uncontrolled in the world population, the more we're taking chance of seeing variants emerging that are going to be even more dangerous than what we've seen with the Alpha and the Delta variant. So I think we really need to have a giant effort of vaccination of the population. I was somewhat comforted, by the way, to see some of the numbers among that. We have now vaccinated between 3 and 4 billion doses worldwide. So the first billion took 20 weeks to be administered. The second billion dose took 6 weeks. The third billion dose took 4 weeks. So it is a unique result in the history of vaccination of humanity, but it's not quite sufficient. I guesstimate that to achieve something like 80% vaccination with 2 doses, you would need somewhere between 12 and 13 billion doses. So it's going to take some time. And during that time, of course, the virus may well spin out other variants that can be even more problematic than what we have to deal with. The worst case scenario being a variant that would escape current vaccine. Now, an advantage of the mRNA technology is that you can very quickly rejig a new vaccines with the new sequence in the mRNA to give a booster shot, but you have to revaccinate everybody again. And so if this is something that could be avoided, it would be much better. So we're touching a little bit the mRNA booster. It's not clear right now that you need a booster for the Delta variant. It's not clear that a booster with the sequence adapted would be better. Maybe, maybe not. We don't have the data. And I've alluded a little bit about the duration of immunity confer with the vaccine. That is also not clear at this time being. So at this point in time, there is no clear indication that a booster is necessary or will be required, but this requires that we keep an eye on how the situation evolve.
Ray, I'm still willing to help you out with reading the questions to give you a break. Val had asked one, but the next one says that studies indicate the vaccines are safe. someone mentioning that you use what long-term studies have been done. And of course, we know timelines are challenging with such fast-moving everything. In occupational health, we know that most things aren't tested for these effects, and that's what leads to diseases and deaths that we work so hard to prevent.
Right. So the thing is that we've been doing vaccination for a very, very long time, and we do not see these kind of long-term sudden disease with a vaccine that we say are possibilities. The thing is, the way that I like to think about it is that what the vaccine does is trigger an immune reaction against the spike glycoprotein of the virus. This is also going to happen with the viral infection. It's just that if you trigger that immune reaction with a vaccine before, it will protect you from exposure to all that the virus has to offer, spike glycoprotein and replication of the virus and effect of the other viral gene. So all the data that we have right now is that it is considerably safer to take the vaccine rather than to chance the mortality and the morbidity with the virus, which we know exists, whereas we have not seen any long-term or any significant long-term morbidity with the vaccine, and there are no reason to think that it would occur. And so if you want to make a rational bet, it's overwhelmingly in favor of taking the vaccine rather than chancing the virus.
Thank you. So I'll just shout out to the crowd because we have no other further questions in the chat. So please raise your hand or unmute if you have any burning questions before we pass it over. Thank Raymond. And I heard this morning on the radio, Israel has started administering booster shots. There's a comment. So there you go.
Yeah, well, the booster shot is being discussed a lot in the scientific community. So on the one hand, you had this one study that I saw showing that the current vaccine are going to have an effect that will last for years. On the other hand, there was a small study done in Israel which suggests that the immunity against two doses would wane beyond 6 months. But some criticism on that study was that it was done on a very small population and may not be true for the long time. And currently in United States, Canada, and Europe, the consensus of public health and agencies and FDA and the like is that a booster at this point is not necessary. What I think may be going on in Israel is that Pfizer is doing a pilot study to show that a booster shot would be safe and would also be effective in that it would provide a boosting of the immune reaction. And but whether it is necessary to maintain the protection conferred by the vaccine or not, at this point in time, it's not clear. It is not impossible. And as you know, there are many people that have speculated that this virus, although it's theoretically eradicable, in practice, it may not be. We may have to do like we do with influenza and have a booster shot once in a while. It's very possible. At this time, however, we don't have the data to say it with certainty either way, but most people think that at this point in time, there is no great evidence for a booster shot being necessary now. Wait and see.
There is a question around personal protective equipment in the chat around safety eyewear and whether or not it is required as a good defense against the virus along with the mask. Is the virus transmitted in this way? I guess ocular.
Yeah, the answer seems to be yes. This is not the first respiratory virus that can be transmitted through the eye. We know from studies on animal that inoculation of the COVID virus in the eye can lead to COVID infection. And so eye protection, in addition to the mask, is certainly a good idea. That being said, so far it appears to be a relatively minor pathways. And so protection of the airways seems to be paramount. There is nothing wrong in protecting the eyes, and indeed it's sensible to have a protective equipment that would block that pathway as well. I would like to emphasize, however, that vaccination is still the most powerful protection against infection by any route.
Wonderful. Well, I think unless there are any other burning questions, that we are out of questions in the chat, Raymond, which just goes to show what a great job you did. And again, we very much appreciate you contributing to this webinar series as you have in the past, and all your hard work. And I did also want to thank my colleague Patricia Phillips, who put this together today, and just it needed our help to get it to the finish line. So it was all Patricia's hard work getting this together, and I appreciate her. And recording and slides will be available likely next week. I would ask Bonnie to confirm that, but I'm thinking next week. And just we have Val's going to speak to us just because we have a couple more minutes about this global summit to end pandemics, and some resources for you to access. So all you interested participants, and Raymond, you're more than welcome to leave or stay, with much gratitude and many thanks.
Thank you very much. It's been a great pleasure for me to present to you today.
Thank you.
Thanks, Ray. And hi, everybody. I just thought, since we had time this afternoon, that I would share a little bit. Many of you would have received an email last week about OCOW and the National Coalition on Aerosol Transmission that OCOW and many in our audience and many others, partners across Canada, have been working on since January, essentially. And we participated in a worldwide event that was quite exciting. So I just wanted to show you some of the content from that, and then we will send out to you the information on how you can access what's currently already available, and to give you a little sense of what the possibilities are for others to get involved. Because let me just share my screen in terms of this this event, which occurred on Wednesday and Thursday. I clicked it. Now I have to click share. Can you see? So this is actually just the brochure in terms of this global summit to end pandemics, which included participation from around the world. Mostly, it was all in English, so mostly from English-speaking countries, but also many European countries, as well as Australia, New Zealand, and North and South America, and then a few Asian participants as well. And it was kind of an overnight thing. It was a bit of an adventure, and Canada actually had one of the two main. There were a series of speakers who they did an opening, which is not yet online, but it will be. It was a 2-hour opening of many high-level speakers from around the world. I'm just trying to, and then they also have done a declaration, which I was going to show you, that they're looking for signatures on. So we will send out a link to that declaration. I can show you what it looks like right now. Actually, you can see people have been in here editing, but very much the pandemic was, or the summit was designed to get all kinds of groups that have been working on COVID prevention around the world, from a lot of different levels, with a lot of different areas of expertise, and with varying degrees of success, to share their successes and their challenges, and to have dialogue about worldwide solutions. That because so much of what we have seen about the disease and its going around and around the world, it has, because people are very much concentrated on national solutions versus international or worldwide solution, and the level of collaboration and even learning from the mistakes or the successes of others has not been as rigorous as we would like, including all of us watching with great, not anticipation, with great dread of what's happening in the UK this week, but also, and in fact, I can show you, and we'll include a link. So I will send a link. This is actually a declaration about about working from a world perspective. They have deliberately called this a World Health Network, being a parallel to the World Health Organization in terms of less politically driven or influenced, and a wide group of community activists and scientists and medical specialists and lots of people who have become engaged at all different levels in trying to make a difference, and have been able to have success in their community sharing strategies. So I will send the link in case people are interested. It's about a 3-page document, and they're looking for hundreds of signatures. I also wanted to show you that there is a link to the organization, which is this World Health Network. And in fact, even since the event, which was Wednesday and Thursday, on Friday, they held an emergency summit about what's going on in the UK. And I can send you the YouTube video for the 1-hour sort of rallying of a bunch of the participants from around the world that was done on Friday after this event. And I also have open here, and I'll just show you briefly because it's kind of just a teaser. The format that was used was for this event was quite unique in that it was using this Oyez platform, which actually had the ability to go and meet people, to go and have so our session, the Canada sharing session, was a special session. So it was in an amphitheater like this, and we could put people down on the stage, and we could have people ask questions, and then all the rest of the people were in the audience, and it was the most virtually real gathering I've ever been to, and it was quite interesting. And they've also created a series of places where people can have offices. So Occ is planning to create an office here where we could in future potentially host meetings with others from the network. And there is meant to be a public link to this website. Right now you can apply to join the network as an organization that's working towards COVID prevention, and that ultimately there will be a public view. And also in the just going back to the main hall, one of the features of the event was that they created, although I don't actually see it right now, there's a world hall right here. Sorry, I'm interrupting. I'll get out of here because you guys are in the midst of something. I'm sorry. I'm just giving that quick tour, but I will leave and share it with others later. Thanks. Sorry about that, you guys. I'm not sure you could hear them, but I actually
No, we don't, Val, not to worry. We didn't hear them. Yeah, not to worry.
I'm trying to leave there so that I'm not bothering them, though, because I could not control my microphone inside there. The other thing I will give you actually right now, though, in this chat is the link to the videos that are currently posted. I was going to show you while I was in the World Hall that a number of organizations, including the coalition, the Canadian Coalition, created 5 or 6 minute videos to introduce themselves and what they were doing around the world. And those are all posted on the hosting organization, which is called the New England Complex Sciences Institute. And there's a series of videos here that are publicly available, including ones that are quite good, I would say. And there's actually one on vaccines and variants, this one right here, that I did. And you can see, I will just pull this up. I won't play it, but you can see how it recorded in terms of seeing the people in the room and who's the speaker and who's the moderator and that kind of thing. So those are available for you to go and enjoy now, and then there will be more later, I guess, basically. And I also wanted to let you know, I think I showed you the emergency summit. The other thing I was going to show you, sorry, the toolbar is covering up my other screen, was that we have posted on the OCAL website the Canadian the hour and a half that was the Canadian presentation. So it was mostly about quite a few people from across the country, from our coalition, from OCAL itself, but also from the Zero COVID Canada organization, and from other activists from Alberta, BC, and Ontario and Quebec, and other places, and then the people from around the world who asked us questions, and the conversation that went on with that. So that's actually already on OCAL's website. So we can include the link to that as well in our follow-up email, but I can also paste it in right now. If I stop sharing, I am going to put this link to the Nexi YouTube channel, and then I will put the link to ours as well. Let me just stop sharing, because I think that's maybe enough of a gist to hopefully you can tell by my voice that it was quite an exciting event for those of us who were there, to see how much is going on around the world, and how many people are really committed to end coronavirus. That's the main, and zero, elimination, or getting it to a level that is, let me just write what this is, I guess, next.
While you're writing, Val, I just want to take a minute to thank you and Kevin and John and Pravesh and others who have really gotten behind, helping workers with this COVID issue, and your tireless nights with the coalition, and bringing us up to speed with all the science. And just wanted to take a minute to do that. I went on one late night and I was on the wrong date altogether. So to support Kevin and Val and John and others. So I just want to take that moment again to thank you all. And feel free to close us out, Val. I'll just let you do that.
I'm just going to put the one more link here, if I can, if people are interested, to the declaration, because they are looking for, there's accepting comments still. It's not completely done, but they don't want edits. They want people to comment on the side with suggested edits, and also signatures to the document that we have to work together as a world. We need to listen to multiple voices. We need to, in terms of professions, and in terms of community impact, and the vulnerable, and that kind of thing. So yeah, okay. And I don't know if anybody has any questions about the event. I know that people have had to leave. That's fine. So we just wanted to share some excitement, and potentially we'll bring some speakers that we met. It was very interesting. I talked to people from Sweden. There are a lot of people from Sweden there, quite concerned about what's happening in their country, and people from Germany, and several from the States, who also sharing their concerns, and but also some solutions. A number of people from Ireland as well. So there will be more in our future events. We may be able to bring some speakers, and as there is a public face and the ability to join the network, we will share that information too. And otherwise, I guess I don't see any questions in the chat, or if anybody wants to unmute and say anything, or Kevin, if you wanted to share your experience of the whole event as well. And it's not just the event, it's the initiative in terms of the level of organization to try and bring all these voices from around the world together to make a difference is pretty inspiring.
Yeah, I guess thanks Val, and thanks Kimberly. I guess the sort of the thinking that still resonates with me after being part of that global summit is some of the conversations that we had with people from other countries. And a case in point, we're talking to people from Germany, and how, culturally accepting they are of wearing better face coverings in public. There seems to be a stronger acceptance of saying wearing N95s, and these are readily available in supermarkets, in shops, and, at really low prices. So it's, they've kind of moved away from cloth face coverings more towards, N95s. I think that's what really stood out for me too. If I could just communicate that. Thanks.
Thank you. Okay, well then we'll say goodbye and thanks again to everybody for attending today, and don't hesitate to email us. Don't please do complete the survey that Vonnie has put in the chat, and um, save the chat if you want those links. But as I said, we will offer a follow-up email that gives some of these links as well to everybody who attended today. And we'll see you next month on August the 20th, I think, again at 1 o'clock.
It is 1 o'clock. Yep.
Okay,
Thank you.
great. We'll see you
And-
all there.
Oh, and I just wanted to quickly add that if you go to the Occ-COVID page that's linked there on our website, by the end of the day, you will find the slides and other information will come soon.
Great. Thanks a lot, Vonnie, and Tony too, if he's still here. Okay. Thanks, everybody. Bye-bye.